Okay, I think I just started the recording. So this is your spotlight session, 8 a.m. On the 27th of January, and I think we're starting. Since I'm kind of like your lab instructor. I think we're starting lab 2 this week, and from my understanding, I did answer some questions about lecture that I kind of want to ask you all. Before we get into lab, but how's chapter 2 going on atomic theory? Do we have any questions about atomic theory or anything relating to atoms or bonds or covalent bonds, ionic bonds, protons, neutrons, electrons? Is there anything we want to bring up there that's on our mind? Students seem to struggle, and you can either chat me or speak up through your microphone, and we'll go from that. Okay. All right. Thank you, April. Still need a little help on polar and nonpolar bonds. Okay. So the difference between polar and nonpolar bonds is basically whether they are able to bind water or not. Okay. If you think about us, us in general, human beings or any kind of living organism, we're pretty much like a bag of water, right? And polar bonds and nonpolar bonds, basically the elementary definition is polar bonds are able to bind water, and then nonpolar bonds do not. Okay, so what would be like examples? Polar bonds, if you want to take like a biochemistry approach to it, like biochemistry would be like that is kind of like what is where, well, it's like biology meeting chemistry, but what it really is what happens in our bodies and how metabolism takes place, and we get energy, and it kind of explains the chemistry behind that. So like glucose, when we take, like, let's say we get like a spoonful of honey and we just eat the honey, right? Chances are that's a type of sugar that's going to get broken down. It's probably sucrose, table sugar. It's going to get broken down into glucose, and then glucose is able to bind to water and get broken down into energy. So glucose is able to polar bond with water and other things, so it kind of helps break it down, and we can use it for ATP or energy. Non-polar bonds, for example, that do not bind to water. It's kind of like, have you ever seen like oil and water and you mix them together and they'll separate out? That's because you have non-polar components or non-polar bonds that will not bind to water, and they actually create what's called a micelle. And it's kind of how soap works. When you wash your hands, basically you're washing your hands with a bunch of non-polar bonds and a solution that will help pick the dirt and bacteria off your hands and trap them in these little droplets. Another thing that polar bonds really do is that it's fat. If you think about fat or oil, it's two different things, but it's similar composition, as in like non-polar bonds. And fat or lipids, if you ever heard of lipids, they are non-polar binding. So they kind of they keep to themselves. They bind each other. They will not reach out and bind water. And we have ways of making them more polar through your gallbladder, and absorbing the good fats and kicking out the bad fats, triglycerides and whatnot. But it really just boils down to polar bonds are bind to water, and non-polar bonds bind basically to themselves. They do not bind water. Does that help answer your question? Okay, I know it was kind of long-winded there. Now, because I got you guys, no problem. That's what I'm here for. I want to talk more about that stuff. If anybody else has any more questions, but before we get into it, I'm putting these up on my YouTube channel. I have all my students, my lab students, like, do you have somewhere where we can watch these at a later date to review the material? I'm like, well, no, but I can figure out how to do it. And so I did it. I created a YouTube account. So all these are going up on my YouTube account, and you're more than welcome to check them out, or you know, trying to listen to me explain something again. And hopefully, if you don't understand it the first time, you listen to it the second or third time, things will start making sense for you. So just want to throw that out there if you're interested. Does anybody have any other questions about chapter 2 or any lecture material? Because I'm kind of tired about talking about lab material. Lab's kind of boring right now. It's a lot of memorization. Okay. Well, we'll just go over some formality things then. So we have a quiz. The quiz is opened up for lab. All right. The quiz 2 is open. Excuse me. Quiz 1 is opened up. It'll close out Thursday at midnight. So if you haven't taken it already, please do. Before we end this today, please remind me to send you all the attendance form. Sometimes I get ahead of myself and I don't send the attendance form, and you guys don't get credit for these, and I need to make sure you get credit. So we got the quiz form out, and excuse me, the quiz on the lab 1 is out. It's a lot of material. It's 20 points. I think there's like 3 conversion questions, 3 baby questions, being the shoulder, cheek, and neck regions of the baby. And if you have me for lab, you probably heard me talk about the different quadrants and regions. I knew those were going to be a few questions on there. So that gives you a large amount right there of the amount of, you know, a good portion of what the quiz is going to be on. Do we have any questions about lab 1? Okay. All right. So lab 2 is microscopy. So microscopy, if we haven't had it already, are we online people, or are we in person? And who do you have for laboratory? In person? Okay. Okay, cool. Are you Monday or are you Wednesday, April? For me. Okay. Well, I'll see you tonight. Holler at me if you come. If you come, we do Zooms too. So if you want to come, you can come in person, but we can always do Zooms. You'll get that email when I send it out around 6:30. Okay, so Garen's got Rainier. Okay. Jaleesa. Okay, yeah, I think I remember you from last week. Welcome back. Okay, and okay, Miss Drake. I think you may get some things from me, Jessica. I share a Blackboard shell with Miss Drake, so she helped me out a lot last year. Okay. All right. So looks like some of y'all, you might have already had this or had time to go through it, but some of you all, I think on Wednesday, we're gonna, this will be kind of redundant tonight, and I'll explain what we're doing. But the memorization part with the microscope, and I'm going to try to share my screen here because I want you guys to see this, what I'm talking about. Let's do here. So now you should be able to see the lab. And the part that I want to talk about magnification real quick. And the magnification right here, the ocular lenses are always going to have a magnification of around 10x, okay? 10x. And so you're going to have, when you're already looking through these lenses, you're going to have a magnification of 10 times. Now, down here are called objectives, and they are also different versions of magnification. You're going to have a 4x, a 10x, and then a 40x. Now, the low power is going to be 4x and 10x, and then we consider the 40x high power. And when you think about it, high power means you're just looking at something a lot smaller. It's going to make it look larger. Okay, so if you have questions in your lab and you'll have questions on your quiz, it's going to ask you what is the total magnification. And the total magnification is going to be what is up here on the oculars, which is 10x. That's like me writing 10x right there. And then down here, these objectives, depending on which one we have selected, 4x, 10x, or 40x, let's say we're going to do the 10x. So if we have 10x objective selected, and we're already looking through the oculars at 10x, 10 times 10 is what? I'm not answering it. You'll have to participate a little bit. What is our total magnification? Chat me something. Okay, that's right. Thank you. So when we write this, you want to make sure we're writing that with the x. We want to make sure we're writing with the x. So now, if it's 40, then we're going to do 40 times 10, which is 400x. And then if we use the low power, which is just the regular 4x objective, we're going to do 4 times 10. And that's considered 40x total magnification. Now, another important thing down here is the condenser. And that's what's underneath the stage. The stage is where we put the slide. And then below the stage is the condenser. And this allows a certain amount of light in and out, okay? And the certain amount of light in and out, what we try to use this for is when we have cells that can be transparent, like epithelial cells. If you're not careful, you can like literally look right through them because you don't see them on the slide, even though you have it focused, but you have too much light coming through the stage, all right? And you pretty much bleach everything out. And what happens is that this condenser, you can modulate it, move it a little bit here and there, and it'll help lower the amount of light coming through the stage. So if you were to lower it, if you were to make the condenser smaller, then that will create less light coming through, and then it gives the cells more contrast, okay? And that way we're able to find what we're actually looking for. We don't have too much light coming through where it just kind of bleaches it out. So that can be important. From Monday's lab, I want to show you all the coarse and fine focus over here. So the coarse is obviously the larger one, okay? And when you use these microscopes in person, you're going to be modulating them, moving them around. The coarse, which is going to move this stage fairly, a considerable distance, okay? And then when we think we get closer to the focus plane, and this will take time to work through this. For you online people, obviously you won't have a chance to do this. If you want to, you can always try to come in person. I know a lot of people eventually have to take medical micro for nursing or hygiene. Excuse me. Let's see. I can't think of any. There's other classes, but medical micro, basically, you're going to have to be able to move, be able to know your way around a microscope. Now, you'll have more chances of working on scopes. But if you feel like you want to start now, or you're just anxious because you want to get hands-on, you can always email me, and you can come to one of my in-person labs, and we'll get you a scope, and I'll show you everything that you need to know about a scope. However, when we get closer to the fine focus, or excuse me, when we get closer to the focal plane, I guess, when we start seeing things underneath the microscope, we start moving the fine focus, and then that'll slowly move the stage up and down. If you think about it, cells are still 3D. They're not going to be flattened out. So when we focus, we're focusing the stage up and down. And what happens there is that we're literally, let me see if I can show you all what I'm talking about. We're looking at different planes. Like if, let's say my head's the cell, right? Obviously my head is 3D. So if we're looking down on my head, we will focus through different layers of my head, or of the cells, okay? And think of it as my head as like a bunch of cells, because these microscopes aren't that powerful to look through like organelles. We're only doing that with nucleus. You can probably see the nucleus because it's stained, and we'll see that tonight. when we do the wet mount. You'll get to see the epithelial cells. So that's kind of interesting. So the stage will help, and it helps move through different planes of the cells. Okay, so there's more to that. We'll probably dig into it a little bit more tonight, but that's just kind of like an introduction of the microscope. Here are on the next page. Let me try to go back to my split screen here. Here are the course functions. Okay, the part functions, microscope functions. So please, you probably want to, you probably want to memorize that, or at least know how to apply them. Here is the condenser that we talked about, and the objectives down here. I'd say you want to highlight these. This is the ocular lens at the very top, where your eyeballs first look into the microscope, and then there's other more important parts to the scope. Now, when we get down here, I think this is taking care of it. These bolded words here, we need to know these for the quiz. That's like a staple throughout the whole class. So when we see bolded words here, that's something that we want to definitely know that answer. Never use the course focus on the high power. That's true. Sometimes you'll break your slide because you've rammed it right up into the objective. So please make notes of these bolded words and these underlined words, because chances are they're going to show up on a quiz or an exam. Now we're kind of getting down to the procedure. And there's going to be certain parts of this that we're going to do. With my folks tonight, we're probably going to be talking a little bit more concepts, and I'm going to give you some examples where I want you to put something in focus, and then raise your hand and have me come by and see that you're able to use the scope. And I think students really like that a lot. And we're going to talk about field or depth of field and the lens effect and what all that means, which is kind of interesting. I'll try to explain it the best I can. Some of it's memorization, and some of it's I could tell a little story, and it probably will make sense. Okay, so let's scroll down through here. This is kind of like where I was talking about total magnification. We're going to go into that in your lab. Here's the lens effect. Let me see if there's other stuff I want to talk about here. Depth of field, we're going to do the thread. Okay, this is a table. This is where we talk about where we're going to do the total magnification. Here's your ocular. Then that's always going to remain at 10x. Okay, always be 10x. And then we'll fill out this graph during our lab time. Here's our wet mount. And then let's talk about cell structure real quick. Okay, this is down here on page 213. Now, on the point that I'm going to try to make here that maybe your other instructors will talk about is that the structure of cells will dictate their function. Okay, and what I mean by that is that basically how these cells look or appear will basically tell you how their function is, okay, what their purpose is in the cell. For the first one is an epithelial cell. Epithelial cells are kind of like more rectangle or more square-like, maybe, not exactly square, maybe like a little rough, but they kind of are large square-like cells, okay? They have a nucleus, and basically, if you think about it, epithelial cells line all the mucosal layers. It's what your skin is, the dead skin, and they act as a barrier, okay? They act as a barrier, and they also act to do other lesser important things, but the take-home message for epithelial cells is that they act as barriers. Now, secondly, cardiac muscle cells. So cardiac muscle cells are more rectangular, okay? They're more like rectangular, and they kind of pinch off at the ends, and they're connected with a bunch of other cardiac muscle cells. So I want to ask you guys a question. Cardiac muscle cells, what's the heart do? Let's just start off simple here. What's the heart do? Chat me this, please. Is anybody there? Okay. All right. Thanks, April. All right. Pumps blood through the body. So when the heart pumps blood, something has to happen for the muscles to contract. Do we know what that is? Like what happens when the heart stops? And we've seen this all in movies. They get the paddles out and they administer a shock, right? And that is electrical conduction. And what happens is that the heart has an SA node in the left atrium, which is the top left part of the heart. And what happens is there's a little pocket of cells called the SA node that will administer a shock, an electrical conduction, or an action potential. And the purpose for these cardiac muscle cells is to conduct this signal throughout the heart that contracts it because it is a muscle and pushes your blood out of the heart. And this is kind of interesting. The way the cardiac muscle cells are designed is that they are designed to carry an electric potential through the heart through very low resistance and contract the heart enough to where it can push blood throughout the body. It's kind of interesting. So I think that's all I kind of want to say there. Now, human blood cells, red blood cells. Does anybody know what a red blood cell looks like? It looks like a donut. Okay, it looks like a donut. If you Google this, yeah. Okay, thank you, Karen. It looks like a donut. And so in the middle of the donut, if you ever were to Google image search a red blood cell, you'll see like a little space in the middle, like a donut would be. That's where your oxygen goes. The whole cell is designed to carry oxygen throughout the whole body. And it's so specialized that this is one of the few, if not the only. Does anybody know what's special about red blood cells that makes them differ from other cells? If you've had me, I've talked about this. I think I kind of talked about it in my first week. If you don't know, that's okay. But you're going to have a question on this eventually throughout one of your academic careers. You're going to have a question on this. What's so special about a red blood cell? And I'm assuming you guys don't know. Normally, if I was in person, I could like see the looks on your faces, and I would know whether if we're thinking or if you're texting or sleeping or whatever. But I think I have your all's attention. No nucleus. Yeah, you're right. Thank you, Alicia. It's no nucleus. That's right. So if the nucleus pretty much gets sucked out, they are anucleated, which means they do not have a nucleus. If they had a nucleus, they would never have enough room to carry oxygen. So red blood cells don't have a nucleus, which is just crazy. At one point they do, and then it gets pulled out of the spleen, and it becomes a mature red blood cell when it doesn't have a nucleus, and therefore it can carry oxygen. And that's what it is. So their structure yields the function. The structure is designed enough to where it could function to carry out its function, which is carrying oxygen to tissues and carbon dioxide to lungs to exhale the lungs out, carbon dioxide out. All right, lastly, sperm cells. So I think we probably maybe all know the idea of what a sperm cell looks like, right? It's got this head on it, like a triangled head where the genetic material are from the males. It's kind of like this little skinny body, and then we have like this tail that comes by. You need your computer? Here. That's fine. So we have these tails, and the tails are more like flagella, right? And this is kind of what helps propel sperm up the vaginal canal against gravity and against normal vaginal flow. Normal vaginal flow is from up to down, and obviously sperm have to travel up to meet the egg in the fallopian tube and hopefully fertilize the egg. So let's talk about organelles real quick. Organelles, what cell organelles are basically is the endoplasmic reticulum, the Golgi apparatus, the nucleus, the ribosomes, et cetera, et cetera. Do we know what the powerhouse of the cell is? Do you all remember what the powerhouse of the cell is? What organelle is that? Yeah, that's right, April. It's mitochondria. So I kind of introduced this when we first talked about the biochemistry part and ATP and energy, right? You know, how we use ATP is the energy currency of the cell. So what mitochondria do is they create a bunch of ATP. And what the sperm cell does with this ATP is that it helps drive the tail. It helps spin the tail to travel up the vaginal canal into the fallopian tubes. So it's safe to say that sperm cells are jam-packed with mitochondria. They have a large number of mitochondria compared to other cells because it is necessary for them to produce lots and lots of ATP to drive the flagella so they can meet their goal, which is fertilize the egg in the fallopian tube. And it's hard work, and they have to, be good swimmers, so to speak. So once again, this is another example of how structure yields function, how the whole structure of the sperm cell and what they look like, their anatomy, i.e. the amount of mitochondria they have, helps propagate them up the vaginal canal and into the fallopian tube. Okay, those are 4 examples of how different cells can yield, carry out their different functions in the body. Now, so let's go over cell structure. That is more memorization there. Be sure you know the difference between rough endoplasmic reticulum and smooth endoplasmic reticulum. There's a Golgi apparatus there that is important, and be able to pick those out. I think last year the question was centrioles. I think centrioles was the one that we asked last year. I'm sure it'll be different, but that was a rare. Centrioles is kind of a rare one to pick out. So make sure we don't skimp on that. All right. So I think that is all we're going to cover today. Do you guys have any other questions for me? That's pretty much an introduction of laboratory. Is there anything in there, anything about lecture? Any more lecture questions? Atomic theory can be kind of tough. Usually students struggle with atomic theory. Protons, electrons, neutrons, we're okay on that. Isotopes, we had conversations about covalent bonds and ionic bonds. Sunday evening, we had a good discussion there. Okay. All right. So let me get you the attendance form real quick. Yeah, Alicia, I'm going to post this. I got a YouTube account now. So if you want to Google or go on YouTube and look for my last name and like put in bio 137, give it a couple minutes to upload up. I'm probably going to have it up here in the next half hour or so, surely by 9 o'clock. So if you want to review it, go right ahead. I think that's how you can do it. And if you can find it, just email me and let me know. All right, let me find the email or my attendance form here. Here it is. All right, so this is the attendance form right here. And I'm going to hang out and have you guys try to fill this out and let me know if you have problems. And then when you got it finished, you're free to go. If you got any questions, let me know. Some people can get it really quick, and then some people got to get hung up on a few questions on there. You can put Wednesday morning with Mr. Mattmiller. You know, you can put the date on there. By 1:37, you can do that too if you'd like. Yeah, Garen, you're good to go. Check back in another time. Okay, thank you. Alicia, probably around 6:30. I'll be there. I'll be showing up in person. I know we got some weather coming in tonight, so be careful. Now, if you wanna, if are you one of my students, or are you gonna be are you somebody else's? I forget what you told me, if you're one of mine or not. Oh, you're online. Okay. All right. So yeah, it's Newtown campus and the SEC building. That's room 303, I believe. And so if you're coming to do like some hands-on, then you can come around 6:30. Obviously, the Zoom won't help you, so I'm assuming that's why you're coming in, is because you want to come get your hands on with some microscopy. So yeah, just email me real quick if you want me to put in writing. I just got off work and I'm just wiped out, so I'll probably be crashing here around 9:30. And I'll probably wake up right before lab, so I won't be able to check my email. Here, I'll just tell you here. Sorry, I'm making this too hard. Newtown campus, 6:30 p.m. I'll get there like right at 6:30, so you'll probably see people out in the hallway if people come. They went. I feel like they'll be doing more Zooms as the semester goes on. All right. See you later. Thank you. I'm going to end this.